Hello everyone. We are now going to go over the peripheral blood flow cytometry plots from a 46-year-old man who presented with massive splenomegaly and atypical lymphocytosis. So this is a peripheral blood specimen, so we expect a mixture of cells, and in CD45 versus site scatter, we see a significant population of lymphocytes that have bright CD45 and low site scatter. Pretty much nothing in the monocyte gate, and in the granular site gate we have 55% of the cells which have high site scatter and dimmer CD45 expression than the lymphocytes and monocytes. And when we track our lymphocytes, we see that 23% of our lymphocytes are B cells. They're negative for CD10, they're negative for CD34. They all seem to be kappa. They are negative for lambda. So these are kappa restricted B cells, and they express CD103. So CD103 is a hairy cell leukemia marker, and that is positive, and they are negative for CD38. So we have a kappa restricted B cell population that is expressing CD103, and hairy cell leukemia and hairy cell leukemia variant will express CD103, and they also present with massive splenomegaly, and they'll have atypical lymphocytes that have these hair-like projections on their cytoplasm, which are pretty hard to see sometimes, but you can see them. And these cells also, have a little bit high site scatter because of their cytoplasmic projection. So they might even go into the monocyte gate sometimes, and they express very bright CD20. And hairy cell leukemia is associated often with monocytopenia. So very often you will see very scant monocytes, or sometimes even no monocytes. So monocytopenia is something characteristic for hairy cell leukemia. Actually, there may be cells in the monocyte gate which are probably just the hairy cells that are extending from the lymphocyte gate into the monocyte gate. They typically have monocytopenia. Monocytopenia is associated with certain neoplasms. The most characteristic ones are hairy cell leukemia and AML with translocation 821. So these are the two conditions that you have to remember are associated with monocytopenia. So now when we have these hairy cell marker 103, we usually go and add a hairy cell tube. Most labs usually have a specific tube for when they suspect hairy cell leukemia, and that has markers like CD25, CD123, 11C, which are, the hairy cell leukemia markers. So we'll see that later, and we have one more tube to go through to go to that. Here is our second B cell tube that has CD5 and CD23, and here we see that our B cells are negative for CD5, they're negative for CD23, they're positive for FMC7, they're positive for CD200, and they are kappa restricted. So hairy cell leukemias are positive for CD200, and variant, hairy cell leukemia variant, is often negative for CD200. That is something that can help. You can remember that. Now let's go to our add-on tube, and here our population of interest is painted in pink. CD22 is also a B cell marker. So this is and CD19, the B cell marker. So that's what they are using to isolate our B cells. So our CD19 positive cells express CD103, and they express CD25, they express CD22, they express CD11C, and they do express CD123. It's a little partial here, but they are positive. So this is classic hairy cell leukemia, and the markers that are positive here that indicates it is hairy cell leukemia are CD103, CD25, CD11C, and 123. So these are specific. None of these are actually specific, but these are all associated with hairy cell leukemia. And hairy cell leukemia has very bright CD20, and it is associated with monocytopenia. So these are the things you have to remember for hairy cell leukemia. Now, hairy cell leukemia variant will also be 103 positive, but it will have some phenotypic differences from hairy cell leukemia, and we will review that in the next chapter. Thank you.